Okay, thank you very much. Here's my company, Cedars Sinai Medical Center. We're a large corporate hospital, private hospital, 1,000-bed hospital, 12,000 staff, dedicated to both looking for cures for disease and treating patients. So the disease I'm going to talk about is Lou Gehrig's disease, or ALS. We've talked about it actually in the previous session. This is the one you don't want to be diagnosed with. There are no cures, no treatments, death normally within 3 years, and it's a paralysis syndrome where you have twitching muscle, which then becomes deinnervated and dies. Most cases are sporadic, but there are a few, as we heard in the previous talks, caused by mutations in single genes. The SOD1 gene, overexpression of that gene causes ALS. Some mutation in the gene causes an abnormal protein. So the approach we've taken in this combined gene therapy and stem cell therapy program, which is funded by CIRM, I'll get to in a moment, is to look at the entire nervous system, which includes astrocytes, motor neurons, and muscle. And what we think happens in ALS is not only do the motor neurons withdraw from the muscle, but the astrocytes around them, these support cells in the CNS, die. So the idea is to replace those support cells that die around the motor neurons. And through replacing the support cells, they will then provide support to the dying motor neurons. So this is a sort of indirect therapy targeting the support cells. And at the same time, we've engineered those cells, a combined gene therapy product. We've engineered those cells to release a powerful drug called GDNF. This is a growth factor that stops motor neurons from dying. So not only do you have a fresh new cell in the spinal cord around the dying motor neurons, but they're engineered to make a product. So this is a combined gene therapy approach, as we just talked about. The cells we've been using are the fetal derived neural progenitors from the cortex. They grow for up to 70 population doublings, and they transition from making neurons to astrocytes, and we take them at around 20 population doublings. When you put those in the spinal cord, they make astrocytes primarily. There are other companies in this space, Neuralstem, Stem Cells Inc., and Q Therapeutics, trying to do similar things. Different product, I don't have time to tell you about those products, but none of them are engineered to make GDNF, and some of them have a different profile. Some make neurons, others make astrocytes, but we're the only group doing long-term cultures, and we use this unique chopping method, so the cells are never dissociated, they stay in contact with each other. And we think that's important when you put these cells into the brain, that they haven't been dissociated in cultures. As a chopping method that we developed many years ago. So we also engineer these cells, and we started with Patrick Aebischer, and now we do it clinical grade with lentivirus. The lentivirus goes in, engineers the cells to secrete GDNF, and these cells will secrete GDNF forever. It's incorporated into the DNA, so these are now GDNF secreting cell line that we can put into patients. ALS is a complex disease. You have a lot of choices if you're going to inject the cells. You can go into the lumbar cord, and these patients are affected in all these different areas. The cervical region, which affects breathing in the arms, the muscles are affected, and the upper motor neurons are affected. So we have a lot of choices. We've focused on the lumbar spinal cord. FDA told us, actually, to focus on the lumbar spinal cord because that's where they want to see safety and efficacy data. So CIRM were nice enough to give us $17.8 million around 2 years ago, in a 4-year program to get to the clinic. I just told you the hypothesis. We're producing the cells with Larry Kuchar at the City of Hope. We got the clinical grade virus from NGVL when it existed, Ken Cornetta. And we hope to be filing the IND in a 2-year period from start. So 1/2A design, I'll get to that at the end, collaboration with Niels and MGH. And the clinical site will be Cedars Sinai with the neurologist Bob Balow and the neurosurgeon Chirag Patel. This is our program. We want to start with the lot production of the cells, then we do the dose ranging studies in animals, and then we file for the IND. Cell production has not been straightforward. This is our biggest challenge. And I'm happy to say 4 weeks ago we froze down 1,200 vials of final product. These have been through the whole manufacturing process. They've passed all their tests. These cells secrete GDNF. We have 5 million cells per vial, and it's a freeze-thaw protocol. So we take these vials, thaw them at the site, clinical site, and the neurosurgeons will inject them into the patients. They're karyotypically normal, and they express good levels of GDNF. Dose ranging, the rest of the studies to get through preclinical studies were first to do the dose ranging, then we actually tried SPIO to label the cells. I'll tell you about that very briefly. Then the safety tox studies and the tumorigenicity studies, which we're going to do at Biotox starting December 1 this year. And finally, mini pigs, we had to use pigs, FDA required large animal. The dose ranging is done. Very briefly, we used 100,000, 500,000, 1 million, or 2.5 million cells dosed into unilateral side, so just one side of the lumbar region of the spinal cord. And I'll get back to that for the patient trial, why that's important in a moment. And then we evaluated the animals. We can do SPIO imaging, so we can actually see here, we did 5 sites in the spinal cord designated by the arrows here. You can see very nicely a unilateral implantation of the cells using MRI and SPIO. The problem with SPIO for us is if you kill the cells, so we actually, where that red arrow is, we killed the cells with a drug, or we just remove cyclosporin. The cells died, but the SPIO remained. And so when we looked at the animals, the animals had no cells surviving, but they still had good MRI signal. This is a problem with SPIO labeling. It gets leaks into surrounding cells, so you can't really tell if your cells are alive or not just based on SPIO, because they could have been taken out, the SPIO, by surrounding cells after they died. So we didn't incorporate this into our final clinical design, because it wasn't going to tell us whether the cells survived. It's useful for the targeting, but not for survival. These are amazing cells. They survive beautifully in the spinal cord. Every dose we tested has surviving cells. Here you can see them in red as a human marker, migrating around the green big cells there, which are the motor neurons. They migrate around the cells. They survive for long periods. We've done, in this set of animals, we did 320 animals at the different doses. And this is summarizing a lot of different work from Jen Gowing in particular in the group, who headed up a team of 6 people. But now we look at onset over one side, and then at the end point of the animal's disease. The animals died about 180 days. And you can see we get a nice dose response in both the number of cells that were implanted with the dose. And interestingly enough, we see more Ki-67 labeling, which is an indication of proliferation at the lower dose. So there's more room for the cells to proliferate. But the proliferation levels go down over time, just as we've seen in every other transplant we've done with these cells in different models. So they don't cause tumors, and they don't proliferate for long, but they do proliferate a couple of times after you put them into the spinal cord. This is a feature of these cells. They make GDNF. We analyze GDNF levels in the different parts of the cord. Here in this panel here on D, you can see lots of GDNF staining around where the cells are. On the other side, in blue, there is no GDNF staining at all. So they're specific to the site where you transplant them, and they release GDNF. And the GDNF is dose-related as well. So the more cells you put in, the more GDNF release there is. So you're dosing the amount of GDNF you're giving to these animals as well. So as I mentioned previously, we'd like to be able to regulate this level so we can turn on and off GDNF expression so the doctor can decide do they need more GDNF or not. We are working on that. But not necessary for ALS. When you're only going to live 3 years, the side effect of GDNF is not as bad as dying. So we're confident with ALS that even if there were a side effect, if the patients lived and survived 10 years, we'd be extremely happy. And so we'd be able to deal with any side effects. But for Parkinson's, it's different because they can live for 20 years. There, if you had a side effect, it may be very significant. The remarkable thing with these cells is the effects they have on the survival of the motor neurons. We get a 6-fold increase, this is a ratio here, of the transplanted versus non-transplanted side. 6-fold increase in motor neuron survival, and that's our efficacy data for FDA. These cells are very potent. They keep the motor neurons alive for long periods of time. And the animals don't get any worse. There's no bad effect. But as we've reported before, the motor neuron survival does not correlate with connection to the muscle in this very aggressive model of ALS. And so we, for the safety data, we're looking at do the animals get worse over time? These are 2 different scores. And the answer is no, that has no adverse effects. We published recently long-term survival, so now we put these into immunosuppressed animals for 6 months. We don't see any tumors, we don't see any nasty things happening, so we're confident these cells can survive a long time and release GDNF. And in the mini pig, we're now pushing ahead doing the preclinical studies for delivery. And we've been using, I collaborated with Nick Boulis for many years, where he uses this frame, as you can see here, which is a frame that attaches to the patient, in this case attaches to the pig, where you can get good targeting. But it's a bit of a Meccano set. Some of the neurosurgeons are a little freaked out when they see this come into their surgery. So we've been developing a new delivery device independently at Cedars Sinai to be used in the CIRM trial. And this is now just finishing the patenting issue, and the ISO as being completed. But this new delivery device is minimally invasive and allows the neurosurgeon to use a simpler approach to inject the cells. We've had no adverse effects with this device. We can get good targeting, and this is a pig spinal cord, and at the bottom it's showing we've delivered GDNF and the cells to this pig very efficiently and very accurately. The final part is the clinical trial. It's an 18-patient trial. The unique design here is we're going to do unilateral injections of the cells. So if we go into the right side, that should be expected to only affect the right limb of that patient. The neurosurgeon will know which side he put the cells in, but he's going to randomize it. The neurologist will not know which side had the cells. So we're going to look for decline in the leg that had the cells versus the leg that didn't. This is a unique aspect of stem cell trials if you're targeting one area of the CNS. You can look at localized effects. And if you get ALS, if it's rapidly progressing in one leg, it'll rapidly progress in the other as well. So you can use the other leg as a control. Now, of course, we have a crossover design. If it's remarkable and the leg has stopped moving, stopped declining, we can then go back in and do the other side. So that's the crossover, the design. So this is the first in man. We are partnering with a company called uniQure, who have the patents for GDNF released from cells, and they're interested in partnering with us to move to the next stage, but we will be looking for other partners. My view is of patents and partnering is if you get something working, then you go get the partnerships, set it up as well as you can beforehand. But I think we'll be able to easily take this through if we show some effect in this first trial, because it's a phase 2A with the bilateral approach. We're on time now for filing the IND in August 15. We had to do 6 months in-life toxicology studies, which are starting December 1. FDA require that to show there's no tumors for 6 months. But once we're through that, the pig stuff will be done. We'll be able to file the IND. So we're very excited. We're not the only people in this space, but we're the only people using modified cells releasing a growth factor. There's 2 trials, one in neural stem that has been doing a trial, and actually shown in 32 patients, they can put stem cells in the spinal cord and cause no adverse effects to the patients. They're still trying to work out if it's actually having an effect or not, but at least they're safe. And there's a number of other companies in this space. Brainstorm is doing intrathecal mesenchymal cells. We really don't know the results from that yet. And there are people hanging bags of mesenchymal cells and infusing them into ALS patients in a few studies, one in Rochester. So we'll see if anything happens there. I doubt whether that will work, given the aggressiveness of this disease, but it may have a modulating effect. We're also approaching muscle delivery to get more clinical effect with a DoD grant of AAV5 GDNF to try and enhance the effect further in animals. And of course, there's the apomotor neuron that nobody talks about. We've got a paper coming out now, just accepted last week, showing that the apomotor neuron is crucial, and we're doing some transplant studies there, and knockdown of mutant SOD1, to show that the apomotor neuron may also be crucial. So we can't think of lumbar in isolation, but I think once we get an effect there, we can then utilize this technology to put cells in 3 different places, and it would be muscle, motor, and lumbar, and of course cervical. So we have a lot of work to do, but we're very excited to get this first part off the ground. This is our flow with Regenerative Medicine in the middle, but we also have a lot of interest in other disorders at the Regenerative Medicine Institute. We've been working in Huntington's disease and shown these cells can modulate there as well. Retinitis pigmentosa, these cells migrate in the back of the eye and protect even without GDNF. And in stroke with Gary Steinberg, we've shown these cells can go in and actually increase plasticity after stroke. And I really started this work with Parkinson's, and again, it comes back to why we haven't moved forward. And the reason is, for diseases such as Parkinson's and stroke, I think we have to, as a community, not just put a cell in that's going to make a drug forever. We have to be able to regulate it in case there's a side effect that we hadn't predicted. So that's my own personal view. We want to get away. These were fetal-derived cells. We've got techniques now where we can take iPS cells using that unique chopping method and make what we call the easy spheres directly from iPS cells. And these easy spheres are stable, expandable system. And we've now, just in Jove, if you want to see, we've got a GMP way of growing out these easy spheres in the same way that we grow out the neural progenitors that I just talked about. And very recently, just come out again a few weeks ago, we've taken easy spheres and made them, using a selective process, into the same neural progenitors that we get from fetal tissue. So now from iPSCs, less controversial, we can get the same cells that we got from fetal tissue, and these cells survive beautifully after grafting. They're almost identical to the neural progenitors I just showed you. So ultimately, we'd like to move to away from fetal tissue-derived iNPCs and into iPS-derived iNPCs. And because we don't have patents in either area, we're very flexible. So we'll go with the cell that works best, and makes most sense and is most ethically sound. But the trial that we're doing with CIRM will go ahead with the fetal tissue. The one caveat with iPS-derived cells is we don't know if they're exactly the same yet until we run them through all the tests or are as good as fetal-derived progenitors. So that's it. To conclude, I think stem cells will... Oh, that's definitely... I was going to show a conclusion slide. I have to... Oh, did I go on too far? Could you put the last slide up just for the conclusion? Thanks. We think stem cells are going to be useful for clinical therapies. The idea is to put the stem cell in to modulate the environment and help protect cells in the brain, but also in spinal cord, but also arm them to release this growth factor, GDNF. So combining the cells with the growth factor, I liken this to putting the troops in, that's the cells. When you give the troops a weapon, that's GDNF, then they're going to fight when they get in there and not just be killed by the environment that they see. And I think they're unique in the delivery can be specified. You can put the stem cells in one place of the CNS and see if it affects that part. And then the patient can serve as their own control. You get a lot more power in your clinical design. And slow but steady may win the day. I think well-designed trials with ways to at least get some mechanism or efficacy from your clinical trial and come out with something is better than just throwing cells in and hoping. And that's it. I'd like to thank the group. And I'd particularly like to thank CIRM for their continual support. And without that, clearly we couldn't have done any of this work. Thank you very much.